So we've already discussed the first parts of glucose breakdown in order to make ATP, right? The catabolism of glucose. The first part is you take that 6-carbon glucose and you crack it in half, and that breakdown of glucose is called glycolysis. It happens just in the cytoplasm of the cell. It produces a net of 2 ATPs, right? We spend 2, we get 4, but it's a net of 2. We also get 2 NADHs, which have high energy electrons, and then we end up with these 2 3-carbon molecules left over called pyruvate. Well, what did we do with those? We took them into the mitochondria, the powerhouse of the cell, where first we cracked a carbon off of each to make an acetyl group. Each acetyl group was bound to a coenzyme A, and we have acetyl-CoA, which then shuttles that acetyl group into the citric acid cycle. And in the citric acid cycle, it is bound to a 4-carbon oxaloacetate, and it goes through the cycle, a bunch of intermediates and enzymes, and we crack off 2 of those carbons, and in the process, we make 2 more ATP, and we've also released a bunch of high-energy electrons. 6 total NADHs per glucose, 2 F8DH2s, which are also high-energy electron carriers. And we've lost all those carbons along the way. As we popped them off, we've lost them as CO2, and they're gone. And so the carbons of that original 6-carbon glucose are kaput, right? We've breathed them out. But there's still a lot of energy left. And there has to be, because so far we've only made 2 from glycolysis, 2 from citric acid cycle, a total of 4 ATPs, and I've promised you over 30. So we need to take those high energy electrons somewhere. And we take them to another part of the mitochondria, the inner membrane. Mitochondria has 2 membranes. The inner membrane is where this happens, and this is called oxidative phosphorylation. Now this is a term that can be confusing, but oxidative just means it refers to oxidation, which is the movement of electrons from one molecule to another. Oxidation is when one molecule grabs electrons from another. So we're talking about the movement of electrons, and we're moving them from one enzyme to the next, to the next, to the next, to the next in this electron transport chain. So again, these electrons are high energy because they are far away from the nucleus, right? They've been juiced ultimately by the sun inside of a plant during photosynthesis, but that's, a while ago. But they have high energy, and as they get passed from one enzyme to the next, boom, 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 they get dropped to a lower and lower energy state. But as they do that, they release energy. Cool. Well, what do we use that energy for? Well, we use it directly to pump hydrogen ions into this intermembrane space. So that's this stuff up here, right? This is all intermembrane space. This is this zoomed in. And that intermembrane space becomes full of hydrogen ions. And because we know all about diffusion, we know that when you have a whole bunch of something on one side of a membrane, it wants to move to the other side, back here into this matrix, right? But this doesn't just want to go over there because of the concentration gradient. It's not just because it's crowded. It's because they're all hydrogen ions. They're all positively charged, and like charged ions repel one another. So they don't just want to move because it's crowded. They want to move because they hate each other. And there's this huge potential energy stored up in here because when we dropped these electrons in energy level, we pumped all these hydrogen ions in here. Well, there's only one way for a hydrogen ion to move across this membrane back into the matrix of the mitochondrion, and it's through something called ATP synthase. And as you might have guessed, ATP synthase is an enzyme, which ends in ase, right, that synthesizes, wait for it, ATP synthase. It does that because it's also a channel, and those protons or hydrogen ions can flow through there, and the movement of ions, electricity essentially, right? That movement of ions that flow literally turns a rotary motor. Imagine like a hydroelectric dam with a bunch of water on one side, and the water is only able to get through this little hole. And you have a wheel that water turns as it flows through. That's really what's happening here. And as that wheel turns, it takes a phosphate and it crams it onto an adenosine diphosphate. And what do you get when you take an adenosine diphosphate and you cram a third phosphate onto it? why you get adenosine triphosphate, or ATP. And this is where we make the vast majority of our ATP. So we can do this as long as we can keep taking those high energy electrons that we got during glycolysis and that preparatory reaction where we got a couple of NADHs and the citric acid cycle where we got a bunch. As long as we can keep moving them here and passing them through this electron transport chain, we're good. But the problem is, unless you can get rid of those low energy electrons at the end, it clogs this whole system up. And while everyone will accept a high energy electron, nobody wants a low energy electron, right? It's hard to get rid of them. It's like, imagine, and we would never do this in the times of coronavirus, but you had a full Coke and you were passing it from one person to the next, right? A full Coke, unopened, sure, everyone wants that, right? But then the first person drinks some of it, passes it on. It's a little less desirable. But you know what? One person, it was your friend, they drank out of it, still mostly Coke. Drink out of it, pass it on. Eventually, you get to this last person. They take their drink, and it's the dregs. It's mostly backwash. It's gross. I mean, there's a little bit of Coke in there. There's some energy, but ugh. Nobody wants it. The only person who will take it is someone who really, really likes Coke. The only person who's going to take this low energy electron is going to be an atom that really, really likes electrons. Well, we already know of one very electronegative atom from Bio 151 when we talked about the polarity of water, right? Oxygen is very electronegative. So oxygen is the final electron acceptor. It will grab these low energy electrons, and as it binds to them, it will also bind to a couple of these hydrogen ions. Well, when you take a hydrogen ion, which is positive, and you hit an electron, you just get a hydrogen, right? And so we end up with this oxygen plus two hydrogens. And when you take an oxygen and two hydrogens, you get H2O, water. And this is why oxygen is necessary for the production of ATP in our bodies. And it's also why water is produced as a waste product of cellular respiration. Our waste products are CO2, which we breathe out because it can be toxic as it builds up because it makes our blood acidic. And water, which we just, can use. So the water's fine. It just floats around. And that is the electron transport chain. So to review that quickly, in glycolysis, we have a 3-carbon molecule that cracks in half to make 2 3-carbon pyruvates, right? And along the way, we get 2 net ATPs and a couple of NADHs. And those 2 pyruvates move along to the mitochondria where they're prepared for the citric acid cycle by cracking a carbon off, right? That also produces an NADH for each of those. And then these acetyl groups get shuttled in by coenzyme A to the citric acid cycle. And in the citric acid cycle, we get bound to that 4-carbon oxaloacetate, and we go through this cycle, crack the carbons back off, and along the way, we get another couple of ATP per glucose, right? 1 per cycle of the citric acid cycle. And we get a bunch of NADHs, 6 total per glucose molecule, plus 2 FADH2s. And now the carbons are all gone. We breathe them out. We shuttle those high energy electrons here to the inner membrane of the mitochondria to this electron transport chain. We pass these high energy electrons from one enzyme to the next, to the next, to the next, to the next. And each spot they drop and drop and drop in energy level and release that energy that they had before. And we use that energy to pump these hydrogen ions into this intermembrane space. We build up a huge gradient of hydrogen ions, which both chemically want to move across because of diffusion, and also electrically because they're all charged, and we let them move through one thing, and it is the ATP synthase molecule here. And as they go through there, they turn a little rotary wheel, rotary motor, which takes phosphate and crams it onto adenosine diphosphate and makes adenosine triphosphate. And now we have it. I recommend you guys watching this video. It's in your BioFlix and your Mastering A&P. It's also in this PowerPoint, which I have now posted for you. All right, now we are going to break down digestion. So you put something in your oral cavity, and you begin both mechanically and chemically digesting there. You move on to your stomach, where you also will be mechanically and chemically digesting there. But we are moving on to digesting different things like proteins. Then the duodenum, we dump more enzymes together with what's left, and we finish digesting it and absorbing it in the jejunum and ileum. By the time we get to our large intestines, we've broken down and absorbed just about everything. We mostly just have to reabsorb water, but because of the bacteria that are there that can break down cellulose that we are unable to break down, you actually do release some more vitamins and minerals that you can bring across into your bloodstream thanks to those bacteria. So again, this is a general look at what you can, the pathway of food there. So let's start with carbohydrates. Carbohydrates, right, let's say you eat a hot dog bun, right, or a candy bar full of sugar. You're going to crunch it up mechanically in your mouth, and salivary amylase is going to begin to break down those giant polysaccharides like starch into smaller and smaller polysaccharides. And it essentially is active until this stuff has been in your stomach for a little while, because it's active primarily at a neutral pH, and your stomach is not a neutral pH, and so you end up denaturing the amylase. It actually gets broken down as a protein in your stomach. So then no carbohydrate digestion happens in your stomach. You have no amylase. But when you move on to your small intestines, to your duodenum, you make another amylase called pancreatic alpha amylase. And that's in the pancreatic juice that gets dumped into your duodenum. And in duodenum it gets mixed up, and then in the jejunum and then at the ileum to a lesser extent, you mix it up and you break those remaining polysaccharides into smaller and smaller and smaller polysaccharides until everything is about 2 monosaccharides long. We call these disaccharides, maltose, sucrose, lactose, and those get broken down by enzymes that are affixed in the cell membrane of the cells on the side of your intestinal wall that line the microvilli on your jejunum. And we call this the brush border, those microvilli. So these are called brush border enzymes, and they break your last disaccharides into monosaccharides, which are now small enough to be absorbed into your body. Right? We can't digest, I should say, all carbohydrates. Things like cellulose, we can't break down without help. That said, bacteria can break them down. They often will release very, very healthy vitamins and minerals for you, but they also produce some gas when doing so. So sometimes very cellulose-filled or fibery foods cause a lot of gas, right? So, beans, for instance, right? They can often cause some gas buildup, but they are very good for you. Once we've broken these sugars down into monosaccharides, how do we get them into your bloodstream? Well, we literally pump them across the outer cell membrane into the cells that line your intestines, and then they are able to move via simple diffusion, or facilitated diffusion, I should say, into your tissue fluid in those villi, where they are picked up by capillaries. When they diffuse into the capillaries, they are brought directly to your liver because of that hepatic portal system. And your liver processes that stuff by saying, all right, if we have too much glucose, we're going to store some of it as glycogen, which is a polysaccharide. So we'll take those glucose molecules, put them back together, store a lot of them, and hold them in the liver until we need them again when it's been a while since we've eaten. Here is a look at this pumping of glucose into the cell and the diffusion of it out here into the tissue fluid, where it's picked up by your bloodstream and taken directly to your liver. How do we then use this sugar? Well, we just talked about that, right? We take it to our cells and then we break it down by glycolysis, and then that preparatory reaction in the mitochondria, and then we move it into the citric acid cycle, and then we move those high energy electrons that we've accumulated to the electron transport chain in the inner mitochondrial membrane, and we make a lot of ATP. This figure from your book, by the way, is a nice summary of what happens and where you get your ATP and your NADH and your FADH2. So glycolysis, we spend 2, we get 4. That's 2 net ATPs. We get some NADH as well. In that preparatory reaction, we get NADH. The citric acid cycle, we get lots of NADH and FADH2, and this shows you where all those electrons will go and the electrons they will produce in the electron transport system. And then during the citric acid cycle, we will ultimately make 2 ATPs as well.